Now I will discuss with you the transfusion reactions. So these the clinical manifestation of the transfusion reactions results from the massive intravascular hemolysis of the transfused red blood cells by the antibody plus the complement. Transfused red blood cells by the antibody plus the complement. I have already mentioned what is the basic role or importance of the complement system within the human serum. Next comes to the part of the which is very much important. It is the hemolytic disease of the newborn. It is also known as Erythroblastosis foetalis. So the hemolytic disease of the newborn develops when the maternal IgG antibodies specific for the fetal blood group antigens cross the placenta and destroy the fetal red blood cells. Which means, say if a mother is Rh negative and the fetus is Rh positive. Rh means rhesus factor negative and rhesus factor positive in case of the fetus. So first, in case of the first pregnancy, when the umbilical cord has been cut down, then the whole blood will be transfused within the fetal cells. So in this case, the particular of the erythroblastosis foetalis most commonly develops when the Rh plus fetus expresses an Rh antigen on its blood cell and the Rh minus mother does not express. So now comes to the part of the trophoblast. What does it mean? That during pregnancy, fetal RBC are separated from the mother circulation by a layer of the cells of the placenta known as the trophoblast. And during her first pregnancy with the Rh plus fetus, and Rh minus mother is usually not exposed to the enough RBCs to activate our Rh specific B cell. So we know that there are two types of the B cell. One is memory B cell, another is the plasma cell. So memory B cell, when it will, these particular of the memory B cells are important for the further work within the particular of the cell or tissue. So at the time of the delivery, however, separation of the placenta from the uterine wall allows larger amounts of the fetal umbilical cord blood to enter the mother's circulation. It is important, and these type of the fetal RBC then activate the Rh specific B cells, which results in the production of the Rh specific plasma cell and the memory B cell in the mother. So the secreted IgM antibody clears the Rh plus fetal red cells from the mother circulation, but so the plasma cells have been cleared, but the memory cells remain as a threat to any subsequent pregnancy with an Rh plus fetus. Clear? So the hemolytic disease of the newborn caused by the Rh incompatibility in a subsequent pregnancy can be almost entirely prevented by administering the antibodies against the Rh antigen to the mother within the 24 to 48 hours for the first delivery, and these antibodies are known as the ROGAM. It is important, which bind to the any fetal RBC that enter the mother circulation at the time of the delivery and felicitate their clearance before the B cell activation and ensuring the memory cell production, which can take place. So before the production of the memory cells, we have to clear the anti-Rh antibodies from the mother circulation, so there will be no memory cells can be preserved for the future use. And such type of the antibodies which are essential for this type of the work known as ROGAM. It is important.